Assalamu alaikum wa rahmatullahi wa barakatuh. Welcome to today's biology class. Inshallah, we will discuss about chapter 14, that is biotechnology, and today is the first class of this chapter. And today's objectives are, inshallah, we will understand the concept of biotechnology, what is biotechnology and who named it, and classical use of biotechnology, that is the ancient primordial use of biotechnology, and its recent modern approach. And we will also know about step-by-step procedure followed in plant tissue culture process and what are the benefits of plant tissue culture. So by covering today's lesson, we will also find some key terms. Inshallah, we will come across these key terms over the period of the class. So at first we start from the concept, from the word biotechnology. In Bangla that is called Jibo Projukti. Actually this term comes from by combining two words, bio and technology, and such kind of words are familiar to us. For example, we also know some words, combined words as biochemistry or biophysics, etc. We know these terms from chapter 1. For example, when we talk about biochemistry, that means when we talk about the chemical reactions or chemistry inside a living organism, that is called biochemistry. Biophysics is the application of physics principles and laws and to invent different electronic devices for the benefit of human mankind, so that the diagnostic procedure, the diagnosis of diseases becomes easier. So that is such kind of another word, biotechnology. That means biotechnology is the use of technical processes on living organisms or microbes in order to express new characteristics or to derive useful products from them. That means we are applying different techniques or technologies and engineering them. We are modifying them. So we are modifying different organisms that might be plants or animals, or even they can be microbes. So we are changing them, or we're using them in order to express new features on them. So these features are useful, and or we can produce different beneficial products for us. And this term was named in 1999 by a Hungarian scientist called Karl Erecki. And biotechnology, though the word is newer, about this word has been using for for about 100 years, but the use of biotechnology starts from the ancient primitive time, not less than 10,000 years. So we say that there was actually primitive use or ancient use of biotechnology that is called classical biotechnology, and for about 100 years we are the modern approach of biotechnology here we see. So we can categorize biotechnology into two time spans. So the ancient use for thousands of years, that is called classical. As an example of classical biotechnology, we can say about fermentation. That means the use of different bacteria or fungus or microbes to produce different milk products, for example cheese or curd or different type of traditional foods. So actually they are the product of fermentation, that is anaerobic respiration done by different bacteria. So human know this concept of producing curd or cheese, etc. thousands of years ago. So though they don't know what is the principle or mechanism behind this fermentation process, they used to know how to make curd or cheese. And there was also an ancient use of biotechnology called brewing. That means production of beer or alcoholic beverages. Though that is haram in Islam, but this concept of producing beer or wine comes from the ancient time. several thousand years ago. People used to know how to make alcohol, drinkable alcohol, from different starchy materials or sugar-rich materials like grapes or apples or sugarcane, etc. So actually, they employ different kind of bacteria or fungus to convert this sugary material into alcohol. Fermentation and brewing are the classical examples of biotechnology. But when we say about modern biotechnology, we actually refer to the modern uses, modern approaches taken in biotechnology. For example, after the invention of the structure of DNA and many much advancement taken place in the in molecular biology or microbiology. So from this time period, for about 50 or 60 or 70 years, this time span is called modern biotechnology. So as an example of modern biotechnology, we can say about many things like vaccine production or human cloning or animal cloning. So there are many examples of this, but we will also only talk about two approaches of modern biotechnology in this chapter. They are tissue culture process and genetic engineering. So here we say that tissue culture is actually the plant tissue culture process for the propagation of plants, and genetic engineering is the modification of plants, or animals by changing their gene, by adding or subtracting or deleting any gene from an organism and making any useful feature expressed on them. So let's start with the process of tissue culture. Tissue culture is the process of growing plant or animal tissues providing necessary nutrient media. So tissue culture refers to the process by growing a plant or animal tissue in an artificial condition providing necessary nutrient medium. That means that will be performed not in natural condition but in artificial condition, and different nutrient material, hormones, or micronutrients that will be provided for the necessary growth. And when we say about tissue culture, we say that tissue culture can be done on the tissues that have totipotent stem cell. So what are called totipotent stem cells? Totipotent stem cells are immature cells present in either plant or animals, which can turn into any kind of body cell when necessary nutrients and stimulations are provided. So totipotent stem cells are present in any animal or plant body. They are actually kind of reserve cells and undifferentiated cells which can turn into any kind of cell when suitable nutrients and signals are present, signals are triggered, they convert into specific type of cell. And totipotent stem cells are present in specific regions of the plant body and animal body. For example, we are talking about here about plant tissue culture. So in the plant body, we say that there are two kinds of tissues at first. There are some tissues that are permanent tissues and some are meristematic tissues. So meristematic tissues are the tissues which have the capacity to regenerate, which have the capacity to undergo cell division. So in some parts of the plant body, we can find some regions where meristematic stem tissue cells are present. For example, at the apical top part of the plant stem, at the apical shoot or apical part of the plant stem, it contains meristematic tissues or dividing cells, or at the root tip, at the topmost part of the root, but at the tip of the root, we can also find dividing meristematic stem cells. So these cells are capable of undergoing cell division and to produce a clump of cell or a mass of cell when suitable stimulations and nutrients are provided. So in this chapter we will only discuss about plant tissue culture. We will not discuss about animal tissue culture, though animal tissue culture can be performed, and it is somewhat little bit complex to perform than plant tissue culture. But so we will restrict our discussion only in plant tissue culture. So here we see that inside a test tube a plant is growing under a artificial culture medium. A plant is growing. So we will discuss about some key concepts now. So what is called explant? Explant is the detached meristematic tissue part of a from a selected plant to use in tissue culture process. So we say that in plant tissue culture we produce multiple copies of a plant. We make actually clones of a plant by taking a small healthy part from a plant. So by using this small healthy part, we can make a numerous number of plant bodies using it. So explant is the detached meristematic part or the collected tissue used in tissue culture process. So for this process, we can use either pollen grain or apical or lateral bud or shoot tip, any kind of tissue that is meristematic and that can be used as explant. And we also say and the term that is culture medium. So culture medium is the artificial medium used in tissue culture process to support and provide necessary nutrients for the explant. As you see that this jelly-like part is actually the culture medium used for tissue culture process. And when we say about callus is the growing undifferentiated parenchyma tissue mass produced from explant during tissue culture process. So inshallah we will see how a callus looks like in our future video. When we say about steps of tissue culture, at first we see a figure, and I recommend this figure to draw and learn, and you can use it while answering the question on tissue culture process. And this figure is simple and easy to draw. Here we see some steps of tissue culture process. At first we say that the first step is to select a mother plant, a healthy mother plant, and explant. So actually by tissue culture process we want to create numerous copies of a plant. So the plant we desire to clone, so we at first choose a healthy mother plant, and from this healthy mother plant we can collect a young leaf that is growing, that has meristematic stem cells inside it. So this part will be actually called explant. So the tissue sample used in tissue culture process is called explant. And then number 2, the process is a preparation of culture medium. So this explant will be placed in an artificial culture medium. So we have to make the culture medium appropriately at first. After the preparation of culture medium, we have to sterilize it, and we have to also sterilize the explant, because the culture medium is rich in different nutrients. So that is a very good host for propagation of different bacteria and fungus. So during preparation of this culture medium, some of the fungus or bacteria can enter into this medium from the air or from the apparatus using. So we have to sterilize the test tube or petri dish and the culture medium, so that no bacteria or fungus can remain here, so they cannot grow in the petri dish or tissue culture process. So we have to sterilize it, we have to kill the bacteria or fungus, and we have to also clean the surface of the explant. So when this sterilization process is performed, then we place the explant into tissue culture medium. So we take the explant into the petri dish or tissue culture medium, and then we allow it to grow into undifferentiated tissue mass or callus. So after some weeks we will see that the explant has converted into undifferentiated tissue mass, that is called callus. And from this callus we can divide this callus part and make multiple number of pieces. We can divide it into multiple pieces, and from each piece we can transfer each of the piece into different conical flask or test tube, and from each of the divided part we can regenerate a new plant from this. So we say that from the callus we can divide the callus, and these divided parts of the callus can be grown into plantlets. So they are the plantlets growing inside a test tube. And when the plantlet is sufficiently grown enough, so it can be readily transferred to a pot with the soil, and when it can adapt with the external environment, then it can be placed in the field. So this adaptation process is called acclimatization, and then it can be transferred to natural environment. So they are the main steps involved in tissue culture process. Now we will see a video. So here we start from the number 2 step, that means preparation of culture medium. When we prepare a culture medium, we should keep it in mind that the culture medium should contain all the necessary macronutrients and micronutrients, and it should also contain some sucrose and growth hormones. Growth hormones will trigger the explant to grow into callus or into a plantlet. So all of the necessary things should be provided in the culture medium. So the person is preparing a culture medium by combining different chemicals in specific composition. So some sucrose or sugar is added to provide a carbohydrate source for the growth of the plant. So the culture medium is now liquid, but we have to make it semi-solid. So to convert it into semi-solid state, we have to put some agar powder inside the liquid, so that and it is boiled in a microwave oven, so that when it turns cool, then this liquid solution becomes semi-solid, jelly-like. So the culture medium has turned into a semi-solid or jelly-like state. But this culture medium may contain different kind of pathogens or microbes, so we have to sterilize it. So we keep the cap closed using aluminum foil by wrapping it, and then placing the culture medium into autoclave machine. So there is the autoclave machine in which high pressure steam and high temperature steam is produced. So the steam contains high temperature and high pressure, 121 degrees Celsius temperature and 15 pound per square inch pressure. So at this high temperature and high pressure, the pathogens and microbes present inside the conical flask culture medium, they all are killed, so that the culture medium becomes sterilized. So now we have to place the explant into the sterilized culture medium. So we should also sterilize the explant, the piece of the plant that should also be sterilized. So and this process is performed in a machine called laminar airflow machine. And when sterilization is completed, then the explant is transferred. Then the explant is transferred into the culture medium. So all of the processes are done very carefully and sensitively and at sterilized condition. So he is getting out the explant and opening the conical flask bearing culture medium, and now he is placing the explant into the culture medium. So when the explant is placed, then the cap is again so then so this process, this tissue culture process can be done in conical flask, or it can also be done in a petri dish. So here you see that after some weeks of placing the callus of explant in the culture medium, after some weeks you will see that some undifferentiated tissue mass has been grown, and that is still growing inside the culture medium. So here you see that this yellow cell mass, actually they are called the callus. They are undifferentiated tissue mass. That means they don't have any differentiated part. They don't have any root or stem or leaf. So the tissue mass is still undifferentiated. So we can use this callus and divide it into multiple pieces, and each of these callus piece can be placed in another culture medium to produce multiple number of plantlets. So here we see that from the callus we have grown a new plant inside a petri dish. So this callus pieces has been transferred to another culture medium petri dish, and then that has that remained for several weeks, and after that we see that a new plantlet has been emerged from this callus. But the plants grown in the test tube cannot grow very much further. So at a stage we have to transfer it into soil bearing pot. So that we have to remove these grown plantlets out of the tissue culture process, and we will transfer it into soil. So he is getting out the grown plantlet inside the jar, and he will place it into soil. So the soil should be free from pathogens and should be fresh. And now this plant is very much weak, so that it should keep in some sort of ICU, so that that is provided with very much good soil without pathogen, and that is covered with a polythene bag and provided with some higher amount of humidity, so that this plant can survive in a very much favorable condition. But when it can adapt to the soil condition, then after several weeks, or it can be transferred to the field after acclimatization or adaptation process. So this is the process, the 6 steps how tissue culture process is performed. Now we will discuss about the uses of tissue culture. What are the advantages of it? What are the benefits of it? So from the plant tissue culture process, we can produce innumerable clone plantlets. They are clone actually. They should be identical to the mother plant, and they can be produced in a very much short time from a small explant. So if we can somehow manage a good explant, from this explant we can theoretically produce unlimited number of clone plantlets from this. And we can also produce disease-free and virus-free plantlets because all of the processes done for plant tissue culture is done in artificial and sterile condition. So we can become very much sure that the grown plantlets are disease-free and virus-free. And we can produce plants in any season of the year, because we can perform this lab protocols throughout the year, and that doesn't depend on season, that doesn't depend on weather conditions. So in artificial lab conditions, we can propagate plants at any time of the year, and that is not, and this type of propagation process doesn't depend on the flowering or maturation of seed, etc. And we can also rapidly produce plants whose natural reproduction is absent or rate is low. There are some plants who doesn't flower very well. For example, there is a palm species called tali palm that is endemic in Bangladesh, that can only be found in Bangladesh. A kind of palm, tali palm, it produces flower after 100 years. So at the 100 year of its age, it will produce a flower, and after then it will die. So its natural reproduction is very much low. It only reproduces once at its lifetime. So there are some plants whose natural reproduction is very much difficult. For these plants, we can use this artificial plant tissue culture process. And we can also conserve and rapidly produce different vulnerable plant species. For example, this tali palm plant is a vulnerable plant species around the world. And we can also, and many countries are successfully using this tissue culture process commercially. And for example, Thailand is famous for propagating orchid plants by tissue culture process, and they're exporting to earn billions of dollars. And Malaysia is famous for palm oil trees. Most of the palm oil comes from the Malaysia, because Malaysia at its coastal region, it has planted numerous number of oil palm trees, and from the oil palm seeds we can get a palm oil. So they have propagated a huge number of plants using plant tissue culture process. And in your book there is also an example of jojoba plant, and we are very much familiar with jojoba because this jojoba oil is very much used in cosmetics, because it has some therapeutic purpose, and there is a good skin healer or a moisturizing property it has. So this jojoba oil can be found from jojoba plant. Actually, that is a native plant of USA, California and Arizona desert plant. And this jojoba plant has been reproduced successfully in India by tissue culture process. So you know that in India there is Rajasthan region here. So Rajasthan has a large desert area in there. So to reduce the amount of desertification, to reduce the amount of land of desert, some scientists have planted jojoba plant in the Rajasthan, so that they can mitigate the effect of desertification. They can reduce the amount of land in desert. So how they can get this high number of jojoba plant in India? So they have collected a plant from USA and then reproduced by a large number by using tissue culture process, and then planted it in Rajasthan. And jojoba has some unique beneficial features. For example, it can be used as a lubricant that can survive at high temperature. So instead of sperm whale oil, jojoba oil can be a good substitute of it. And production of different fruits, flowers and crop plants, tissue culture process can be used. So today what we have learned, let's revise. So today we have discussed about what is biotechnology. There is the when we employ different technological approaches on biological living organisms to produce different new features or new products. This concept is called biotechnology, and we have divided the use of biotechnology into two times, that is ancient or classical use of biotechnology, and today is about 100 years modern biotechnology. And we have also learned about step-by-step procedure of tissue culture, plant tissue culture process, and we have also learned about benefits of plant tissue culture. So by this lesson, we have also learned some new key terms. For example, we can define what is biotechnology, what is called plant tissue culture or micropropagation. We have also discussed about pluripotent stem cell, that are the undifferentiated cells present inside the body that can turn into any kind of cell under appropriate suitable signals and nutrients. And we have also learned what is explant, the meristematic tissue mass used in plant tissue culture process, that is called explant. Culture medium is the artificial medium used in plant tissue culture process that contains suitable nutrients and growth hormones. Callus is the undifferentiated tissue mass produced from the explant in culture medium. Sterilization is the process of killing the microbes present in the explant and culture medium, and acclimatization is the adaptation of the new plantlet to the natural environment. So at the end of this lesson, you have a homework that describe the steps of plant tissue culture process. No more today. সবাই ভালো থেকো ইনশাআল্লাহ। আসসালামু আলাইকুম।